So this is what I just said. I forgot I had this diagram, so I drew it all out. Here you can see with the reds and blues, right? The random lining up of homologous pairs makes all of these combinations of chromosomes and the genes on them equally likely. And that was what gave the result of 9 to 3 to 3 to 1 and the idea of independent assortment. Okay, this then is one of the 4 reasons why you are different than your brothers and sisters. Because even though you came from the same parents that had the same chromosomes, the way they lined up in the cell that divided by meiosis to make the gamete that formed you, both in mom and in dad, let's pick those two, the way they lined up was completely random. And with 23 pairs of chromosomes lining up randomly of each other, there's that many different kinds of combinations of sperm cells that can happen in egg cells just for this reason. Now, there's another reason for genetic variability, another reason why you're different than your brother and sister, and we'll get to those later. Came a time in history when Mendel's work was rediscovered. We saw his ideas, but we knew that this wasn't the whole story. We knew, for instance, that sometimes, like in some plants, and we'll go back to flower color, you don't just have 2 flower colors. In fact, Mendel knew this, but it was part of his genius that he chose to try to explain the simple stuff first. He never could explain this kind of stuff. Red flowered plant, white flowered plant, all their kids are pink. Well, we simply call this incomplete dominance. Knowing about genes now, if a kid gets a red flower gene from one parent and a white flower gene from the other, one of them doesn't dominate, the dominance is not complete, right? And the phenotype is somewhat in between the 2 phenotypes, red and white, pink, right? But this pink flowered individual still has a red gene and a white gene. That's what it's going to give their kids. So if we cross 2 pink flowered individuals, F1 cross, right, we're going to get this ratio of phenotypes in the offspring. If we had simple dominance here, we would get 3 to 1. All these kids would be red flowered, excuse me, red. If red was dominant, we'd get 3 red to 1 white. But since we have a different situation in terms of phenotypes here, this red flower gene produces a protein, the white flower gene produces a protein, and when both of those proteins are in a plant cell and light falls on it, they reflect back light that look to our eye to be pink. So that's the chemical basis, the molecular basis of incomplete dominance. So then we have the idea of codominance. And if you notice here, back to our EOC syllabus stuff, they don't have incomplete dominance in here because actually molecularly, gene-wise, and chemically, incomplete and codominance are the same. So I guess they chose codominant to put in here. I'll give you a classic example of codominance and one other thing. That comes with blood types. Our blood types, our ABO blood types, are an example of codominance. And if you called it incomplete dominance, it wouldn't be the worst sin in the world. Okay? Here's the idea. Red blood cells have proteins on their surface. One of those proteins is simply called the A protein. Another protein is called the B protein. And just like we talked about before, you get 2 of these genes, one from your mom and one from your dad. These genes are often symbolized with an I with a superscript, because if you got a gene for that A protein, and a gene for that B protein from your other parent, your red blood cells would have both of these proteins, and you would be said to have type AB blood. That would be your phenotype. That would be the description of the result of these genes, type AB blood. But it turns out there's another gene here, and it's called the O gene. It's symbolized by a little I, which suggests correctly that it's recessive to the A gene. It's also recessive to the B gene. So if you got these 2 genes, you would be heterozygous. You would have 2 different genes, but you would have type A blood. You could also have type A blood by getting two type A genes. You could be homozygous type A. Likewise, you could be homozygous or heterozygous for type B. And then you could have type O blood, which means you got two of these recessive genes. So see, here we have four phenotypes: type A, type B, type AB, and type O. And this is a favorite trait to ask heredity questions about, because it covers lots of bases, including this one. I'll say it while it's here. The fact that for this trait, and in fact this is more often true than not, but we haven't had a trait yet where this was true. The fact that for this trait, there's not just two different kinds of genes. What are different kinds of genes also called? Alleles. There's not just two different kinds. In this case, there's three. There's often more, right? But in this case, there's three. That's an example that's simply called multiple alleles. Alleles are different genes for the same trait, right? So far, we've only looked at traits that had two different alleles, right? Purple flower color, white flower color. But for this and many other traits, there are multiple alleles. But what is not changed, there might be three blood type genes, but you only got two. One from mom, one from dad. That hasn't changed. This is what I just said about that. So now our last thing here in this little unit, which is an exception to Mendel's ideas, is that some traits are determined not just by one pair of genes, but by many, pairs of genes. I would have chosen a different term because polygenic and multiple alleles are almost like identical synonyms, just word-wise. Poly and multiple are synonyms. Genic and allele are pretty much synonyms. But what this word means is that one trait, and human skin color is the classic example, one of many, but the classic example of a polygenic trait. There's at least three pairs of genes that go into determining this one trait, skin color. One trait, more than one pair of genes. Not more than one gene, because all the traits we've talked about, right, were, determined by more than one gene, determined by two genes. Now we're saying your skin color is determined by 6 genes. There's probably more, actually, but at least 3 pairs have been identified, right? But what is true again is that when you make gametes, you have these 2 A genes, you're only given one to your kids. You have 2 Bs, you're only given one to your kids. That idea of independent assortment still has not changed. So here's another very simple, actually, but very likely to get a question about this. What determines phenotype? Well, almost always, not always, but almost always, it's a combination of what genes you got from your parents and environmental factors. Skin color makes this easy to understand, right? Your skin color at any one time in your life has to do with your diet, what kind of stuff you might smear on your skin, but also mainly how much time you spend in the sun. But it also has to do with the genes that you got from your parents in terms of those dark and light, different kinds of genes that we just looked at. So now we have a different way to talk about what parent has this trait and what parent has that trait, to make a chart out of it that's called a pedigree, or like a family tree, right? So you're going to get questions where you're given a family tree, like let's pick this one first, all right? This trait we're talking about is whether your earlobes are attached or not attached, right at the very bottom there. Are they attached all the way down, or are they like not attached all the way down, right? Attached earlobes is a dominant trait, but let's look at this. They use F's to symbolize the genes that cause this trait. I'll talk about that in class. This is not really actual. It's just used all the time, even though it's not true. Okay? So if you're trying, if you weren't told, like I just told you, which of these traits was dominant, and you're looking at this chart where you went to a bunch of people, right? You found out, did you have attached earlobes or free earlobes? And then you made out a chart showing, did you have attached earlobes? We're going to symbolize you as either a box male or a circle females that aren't filled in? Or do you have free earlobes? If you do, we're going to call you filled in, right? We're going to say you have that phenotype by filled in. So if you pretend that these genotypes aren't here, which make it obvious, if you just had this chart, how would you figure out which trait's the recessive one? The one symbolized by the filled in or the one symbolized by the not filled in? Here's what you look for. In this pedigree, in any place, do I have two parents that have the same trait, either both not filled in, like these two, or both filled in? Do I have any two parents that have the same trait and have even one kid that's got the different trait? Well, yes, I do. And here it is. Here's two parents not filled in. They have two kids that have the filled in trait, whatever that is. Here again, I have two parents that not filled in. This kid is filled in. What this means is that the trait symbolized by the filled in, circle or square, has to be recessive. Why? Because if two parents had the same trait and that trait was recessive, all their kids would have to be recessive. They can only have recessive kids if all they have is recessive genes. So since these two parents with the same trait had a kid with a different trait, that means two things. This trait has to be recessive, and it also means they have to be heterozygous, right? Now let's look over here. Let's look for that same pattern. Oh, look, I see it here. Here's two parents with the same trait. This time it's a colored in trait, and they have a kid with the trait represented by not colored in. In this one, this is no widow's peak, right? In this one, no widow's peak is the recessive one. The kid with two parents that are different than it always has to be the recessive trait. Follow, follow?